Introduction. Nanopore sequencing technology has revolutionized the field of genetic analysis, offering unprecedented capabilities for the detection and characterization of nucleic acids at the single molecule level. This advanced technique utilizes the concept of ion channel protein-based sensing, combined with cutting-edge nanofabrication methods, to enable the rapid and accurate sequencing of DNA and RNA molecules. In this comprehensive script, we will delve into the principles, techniques, and applications of nanopore sequencing, exploring topics such as nucleotide sensing, signal transduction, base calling, library preparation, real-time sequencing, protein identification, single molecule analysis, bioinformatics, hybrid approaches, and the challenges and future prospects of this exciting technology. Nanopore sequencing is a powerful method for deciphering the order of nucleotides in DNA and RNA molecules. At its core, this approach involves the use of nanopores, tiny holes that are only a few nanometers in diameter, to electrically measure the passage of individual nucleotides through these pores. By monitoring the changes in electrical current associated with nucleotide translocation, the specific sequence information can be obtained. This breakthrough technique has revolutionized the field of genomics, offering advantages such as real-time analysis, single molecule resolution, and long-read capabilities. To facilitate nanopore sequencing, specialized ion channel proteins are utilized. These proteins can form stable nanopores in a lipid bilayer membrane, creating a pathway for nucleic acid molecules to pass through. Nanopore fabrication techniques involve the incorporation of ion channels into artificial membranes, such as lipid bilayers or solid-state materials. The choice of protein and membrane material depends on the specific application and desired performance characteristics. Furthermore, advances in nanotechnology have enabled the development of synthetic nanopores with controllable dimensions, allowing for improved accuracy and sensitivity. Nucleotide sensing lies at the heart of nanopore sequencing. As individual nucleotides traverse the nanopore, they cause transient disruptions in the ionic current flow. These disruptions, known as blockade events, are highly characteristic and can be attributed to the different nucleotide bases. By analyzing the variations in current amplitude, duration, and shape associated with each blockade event, the sequence of the nucleic acid molecule can be determined. This process involves the development of robust algorithms and statistical models to accurately interpret the complex signal patterns. Signal transduction and data acquisition are critical components of nanopore sequencing. As the nucleic acid molecule passes through the nanopore, the associated blockade events are captured and converted into electrical signals. These signals are then amplified, filtered, and digitized for further analysis. High-resolution data acquisition systems, combined with advanced signal processing techniques, enable the extraction of meaningful sequence information. Real-time monitoring of the translocation events provides the added advantage of rapid sequencing, as the results are obtained in a continuous fashion. Base calling is the process of determining the specific nucleotide bases from the electrical signals generated during nanopore sequencing. This step involves computational algorithms that convert the raw signal data into a readable sequence. However, due to the inherent noise and stochastic nature of the measurements, base calling can be challenging. Techniques such as hidden Markov models and machine learning algorithms are employed to improve accuracy and reliability. Additionally, error correction methods based on consensus calling and statistical analysis are implemented to enhance the quality of the obtained sequence. The preparation of DNA libraries plays a crucial role in nanopore sequencing workflows. Library preparation involves the fragmentation of the DNA molecule, the attachment of specialized adapters, and the amplification of the target regions. Various enzymatic and chemical methods are used to achieve these steps efficiently. Additionally, the introduction of unique barcodes allows for multiplex sequencing, enabling the simultaneous analysis of multiple samples. The choice of library preparation method depends on factors such as DNA source, sequencing objectives, and downstream applications. One of the significant advantages of nanopore sequencing is the ability to obtain real-time sequence data. This real-time analysis enables rapid identification of pathogens, characterization of genetic variations, and monitoring of disease progression. Furthermore, the long-read capabilities of nanopore sequencing facilitate the assembly of complex genomes, interrogation of structural variants, and investigation of epigenetic modifications. The versatile nature of this technology has led to its widespread application in various fields, including clinical diagnostics, infectious disease research, and evolutionary biology. In addition to DNA and RNA sequencing, nanopore technology can be applied for protein identification and characterization. By modulating the nanopore properties and using biological nanopores specifically designed to interact with proteins, the translocation events of individual protein molecules can be monitored. This approach enables the investigation of protein structure, dynamics, and interactions at the single molecule level. The integration of nanopores with other analytical techniques, such as mass spectrometry and optical sensors, further enhances the versatility and sensitivity of protein analysis. Nanopore sequencing allows for the analysis of individual molecules, providing valuable insights into heterogeneity and dynamics that are often overlooked in bulk measurements. This capability is particularly useful for studying epigenetic modifications, such as DNA methylation and histone modifications, which play crucial roles in gene regulation and cellular identity. By coupling nanopore sequencing with chemical modifications detection, researchers can unravel the epigenetic landscape of genomes with high resolution and accuracy. The vast amount of data generated by nanopore sequencing necessitates robust bioinformatics tools and data analysis pipelines. These tools are essential for the accurate base calling, quality control, alignment, variant calling, and annotation of sequenced nucleic acid molecules. Various software packages and algorithms have been developed to streamline the data analysis process, enabling researchers to extract meaningful information from the complex datasets. Novel approaches, including neural networks and deep learning algorithms, are continuously being explored to further enhance the efficiency and accuracy of data analysis in nanopore sequencing. Hybrid approaches involving complementary sequencing technologies have emerged to address the limitations of nanopore sequencing. By combining the strengths of different platforms, such as short-read sequencing and optical mapping, researchers can achieve greater accuracy, longer read lengths, and improved genome assembly. Moreover, the integration of nanopore sequencing with other analytical techniques, such as PCR and single-molecule fluorescence, enables multiplexed analysis, site-specific modifications detection, and dynamic profiling, expanding the scope of applications in diverse fields. Although nanopore sequencing has made significant progress, several challenges still need to be addressed. The accuracy and speed of base calling, reducing errors introduced during library preparation, improving the signal-to-noise ratio, and enhancing the throughput are among the current focus areas. Additionally, the development of robust sample preparation protocols, scalability of nanopore arrays, and cost reduction remain crucial for the widespread adoption of this technology. Continuous innovation and collaborations between scientists, engineers, and computational biologists are key to overcoming these challenges and shaping the future of nanopore sequencing. In conclusion, nanopore sequencing technology has transformed the field of genomics, offering unparalleled capabilities for the analysis of nucleic acids at the single molecule level. This method, based on ion channel protein-mediated sensing and advanced nanofabrication strategies, enables real-time, long-read sequencing with high accuracy and sensitivity. With its broad range of applications in genomics, proteomics, epigenomics, and beyond, nanopore sequencing holds immense promise for unraveling the complexities of the biological world. As researchers continue to address the challenges and push the boundaries of this technology, the future of nanopore sequencing appears brighter than ever.